Okay. All right. Well, thank you so much for having me again. Hopefully this is working. Thanks for having me on to talk about my favorite topic of all time, fungus growing ants. You guys might be familiar with the most common example of this phenomenon. We've got leaf cutter ants. Maybe you thought the ants ate the leaves, or maybe you weren't familiar with any sort of relationships at all between ants, plants, and fungi, and you were picturing something like this. Now, while ants don't farm the full fruiting bodies of agarics like this, they do cultivate agaricaceous fungi. So what that means is they farm fungi that once had the innate traits of a cap stem and gills. So although their farms aren't quite this flashy, the idea isn't too far off, genetically speaking. Fungus-growing ants or atine ants are in the ant subfamily Myrmecini, tribe Attini, and subtribe Attina. There are 19 genera in that subtribe, and I have them color-coded here based on different farming practices. We'll be getting into those agricultural systems a little bit more a little later on here. So atine ants are broken down into multiple different clades that are believed to share a common ancestor. So we've got Paleotini and Neotini as the two most expanded clades. The Paleotini contains more ancient lineages extending back to the establishment of this tribe. They cultivate less specialized fungi that are acquired more willy-nilly. Essentially, they're either scavenged, stolen from a neighbor, or they're passed down vertically from the queen's nuptial nest, which is the nest she was born in. The Neotini encompasses all other genera you can see as that branching web of blue on the chart here. This includes more divergent species that share obligatory symbiosis with their fungi, which means they cannot live without each other. This is the most recent of the two to emerge. Now, these two main clades are broken down furthermore into two additional clades, the lower and higher atine ants. The lower atines are within the Paleotini and the Neotini. They utilize more ancestral substrates in their gardens, such as decaying plant material, wood, insect detritus, being fecal matter, discarded body parts, stuff like that. The higher atine ants are within the most recent half of the Neotini. They developed their own clades of specialized fungi that are found nowhere else but in cohabitation with these ants. And this includes, but isn't limited to, the leaf-cutting ant genera. They have shown enhanced protein digesting enzyme activity within their fungus gardens. So what that mouthful means is their fungi have proven to break down plant material at a very increased rate for their symbionts compared to all of the other fungi. Now you're going to see a very unique example of quadripartite symbiosis, which means there's a relationship happening between four different organisms at the same time. So there's three mutualists who all benefit from each other: the ants, their fungus gardens, and actinomycetes bacteria. And then there is one parasite that jeopardizes the way of life or life in general of the latter three. That's going to be pathogenic fungus Escovopsis. We're going to get into all of these organisms and their relationships to each other as we go on. First, we're going to get into the history, and we're going to talk about what the scientists of the Smithsonian determined when they entered our timeline. Using phylogenomic data, they traced the evolutionary history of these ants and constructed the following timeline and chart. So although this chart is a little bit outdated, in 2017 it was published, they're still considered a holy grail amongst myrmecologists studying atine ants. I don't believe this has been updated, but there have been 4 genera added since this was published. We have Xerolitor, Myceta malarius, Paratrachomyrmex, which were all derived from one Trachomyrmex in 2018 and 2019. And then Amoemyrmex, which is a new leaf-cutting ant genus. Looking at the timeline, we go back 66 million years ago to the KPG mass extinction event. That's when it's believed to have jump-started the behavior of the ants turning to fungus for sustenance. Nuclear winters, so soot, debris, ash filled the air and blocked out a lot of sunlight, which was detrimental to a lot of living things, especially our photosynthesizers. This left the ground covered in a blanket of decaying organic material and lack of sunlight. But what decomposers do we know that like to eat dead plant material and they don't require sunlight? Fungi. So about 61 million years ago, that's when the ants turned to farming it within their nests. That's when the lower Attine ants emerged and began practicing lower agriculture. 34 million years ago, we have the terminal Eocene event, which was a decrease in global temperatures up to a 68-degree drop in winter temps. This expanded drier habitats globally. So around 27 to 31 million years ago, the ants expanded with these habitats, bringing their fungi with them. This is thought to be the evolutionary rising point of higher agriculture because the fungi became completely reliant on the perfectly controlled environments within the ants' nests. The Neogene period within the Cenozoic era began about 23 million years ago, and this marked a lot of important changes, including the expansion of grasslands. Leaf cutters emerged the most recently, 18 to 19 million years ago, and they're very widespread through Central and South America, Mexico, southern US. Leaf cutters have been noted to use a variety of fresh plant material. Leaves, obviously, we got leaves, then we have flowers and blades of grass. Now we're going to discuss the strategies of fungal transportation. So these are the different ways that the ants can bring this fungi to their nests. We have vertical transmission, which I mentioned earlier, which is when a queen elate takes a bit of mycelium from her nuptial nest where she was born, and she starts her own colony with it. Then we have horizontal transmission. This is the main practice of the lower Attini. They find their fungi in nature or they steal it from a neighbor. Now, with transportation, if you're familiar with mushroom cultivating, you know that it can be really easily contaminated by bacteria, pests, molds. So how do these queen ants transport their fungi without picking up unwanted hitchhikers? All ants are equipped with an infrabuccal pocket. Infrabuccal, meaning situated beneath the mouth, is a filtering device that the ants use to filter out liquids from the solids that they cannot digest. These solids are later expelled and put in a disposal chamber or they're fed to their larvae. Here's another look at the infrabuccal pocket. And this was taken right after the ant had eaten. So we can see the liquids traveling down the esophagus and into the foregut in that full infrabuccal pocket. Now the Attine ants, have developed an adaptation. They don't only use this as a transportation device, but they use it as a weeding device. So they will groom their gardens and they will remove any harmful bacteria, parasites, enemy fungi, store it in these pockets. And when the pockets become full, they expel them as infrabuccal pellets. These pellets have been noted to contain minimal or dead amounts of the enemy fungi, which shows that they have been exposed to antibiotics. It's not sure if the antibiotics are present within the pocket itself or if they're using the cultures that live on their cuticles. Here's some pictures of some infrabuccal pellet piles of Trachomyrmex and costatus. They have specific workers that are in charge of transporting these pellets into disposal chambers. These disposal chambers are checked and groomed regularly to make sure that the parasites are not spreading. Now we're going to get into how and what they grow. There is still a lot to be discovered about the fungi that these ants grow. Between name changes or not having names at all, things can get a little bit dicey. So I'll be making sure to repeat a lot of key classifications so we can be sure which family of fungi that we're talking about as we go through each system. Sorry. So first we're going to get into how. We remember this from earlier. You might have noticed that there's 4, maybe 5 different ways of farming. There's a little bit of debate on whether or not leaf cutting is considered an additional agricultural system because it is within higher agriculture, but it's mostly accepted as a 5th system, so we're going to carry on and say that it is. So here are the 5 systems in order of oldest to newest to emerge, and we're going to start with the one that started it all after we get into these type of symbiotic relationships. I'm going to cover these in each one of the relationships we go through, whether or not they're facultative or obligatory. So facultative symbionts are mutualistic, but they're not necessarily exclusive. This means that they live with each other and benefit, but they don't need to survive. And these fungi can interbreed with wild fungi. Then we have the obligate symbionts, which are exclusive. They're completely reliant on each other. They cannot interbreed with wild fungi. Here we're going to get into the what now, the part we're all wondering. What do they grow? So all of their fungi is in the Basidiomycota phylum, class Agaricomycetes, order Agaricales. There are 2 families that they're broken into. The Pterolaceae, which contains 1 out of those 5 agricultural systems, and then the Agaricaceae that contain the rest, the other 4. We're going to start in the Agaricaceae. Going right into that lower agriculture, all of the Agaricaceae family fungi are exclusive to the tribe Leucocoprinae. This chart is a really useful way to see the relationships and the similarities and how they're all connected. So I'm going to show this in between each switch of agricultural system. 61 million years ago, that's when lower agriculture began, when a paraphyletic group of 85 ant species started cultivating. This is different than a clade because it does not contain all of the descendants of the common ancestor, only some. This is an ancestral system that rose simultaneously with the ants and rose in 2 different clades. There are usually, but not always, free-living fungi that they are cultivating, so it is considered facultative. They're found across North and South America, varying in nest size. The clades are clades 1 and 2, a perfect example of that namelessness I was mentioning earlier. They thought that they rose simultaneously, but it was later kind of hypothesized that clade 1 is just slightly older than clade 2 because all of the higher agriculture, more specialized fungi derived from clade 1. They use ancestral substrates in both of these clades, and they can be concentrated further into many different subclades depending on the research. There are going to be a lot of exceptions in lower and higher agriculture, one of them being the Siphelmyrmex wheeleri group. It's a clade of 4 species. We have Siphelmyrmex wheeleri, C. costatus, C. longiscapus, and C. muelleri. They're all known to cultivate a phylogenetically distant fungi from clades 1 and 2. Those are further separated into clades A through G, and that is only utilized in this specific group of ants. Here's a little chart kind of breaking down the branching and how many years in between them that they separated. Really cool to dig into. So here's that chart again. You can see on the left here the lower fungal cultivars, clade 1 and 2 we just covered. Now we're going to stray from the Agaricaceae and we're going to jump into the Pterolaceae just for a moment. This is going to be our coral agriculture. There's only in 2 clades, which are just 2 species, Myrmecopterula nudihortorum and Myrmecopterula velohortorum. These were both derived from the genus Pterula in 2020. Coral agriculture rose about 48 million years ago, not too long after the lower agriculture. This is all cultivated by the Apterostigma pilosum group, closely related but separate clades. This genus does have a third species, but it isn't considered a clade because it isn't always cultivated by the ants. That's going to be Myrmecopterula moniliformis. This is considered an obligatory relationship, and it's believed that it possibly used to be a parasite on Leucocoprinae, and it just evolved to encourage ant transportation. They're found in Costa Rica with nest sizes of 100 to 1,000 workers. We're going to look at clade 1, Myrmecopterula nudihortorum. This is the Apterostigma mani subclade group of ants. This is a lower atine subgroup. This was previously referred to as ant cultivar G4, cultivated in sponge-like masses on the nest floor, which is very similar to the behavior of lower atine ants. They're found in the neotropics of South America with very small nests with 30 to 100 workers. The second clade, Myrmecopterula velohortorum, is cultivated by the Apterostigma dentigerum subclade. This was previously referred to as ant cultivar G2. They're thought to be descended from the first clade, and the differentiating practice that made them evolve separate is believed to be the veil of mycelial threads over their fungus garden. It's thought that the ants they take part in doing this. They take part in planting it like this. So that is the differentiating factor between the two clades. They're also found in the neotropics with very small nests of 30 to 100 workers. That third species I mentioned that isn't a clade, but is just kind of floating within the genus is Myrmecopterula moniliformis. This is found growing out of the top of dead nests or living nests of the previous two clades, and it has bead-like sterile structures that grow out of the top of these nests. And sometimes they have branched coral fertile structures that grow out of those sterile structures. Sometimes this can still be found being cultivated by the ants, but most of the time it's found growing out of the top of their nest. It's thought that the presence of sterile and fertile surfaces means that this was an escapee from cultivation, and it became partially free living. So you can see here on the bottom right, we just went over coral fungus cultivars in the Terralaceae. So now we're going to jump right back into the Agaricaceae to discuss the remaining three systems. Yeast agriculture. Yeast agriculture rose around 32 million years ago in the Siphelmyrmex ramosis group of ants. That's a clade of 19 ant species who grow fungi in a yeast-like unicellular phase, which is otherwise really uncommon in the order Agaricales. Siphelmyrmex ramosis group is split into 5 different subgroups, and they supplement their diet with its nectar and sap, which isn't often seen in atine ants. This also arose from clade 1, just like the higher agriculture. It's facultative because it's believed that these yeasts could survive outside of the nest, but they haven't been found doing so yet. So that one's a little bit debatable. They're native to the neotropics, but can also be found in Florida with 100 to 300 workers per nest. The way that they cultivate their yeast is this ant here in this picture is carrying a ball of caterpillar frass or caterpillar poo, and it's taking it to create a ball of substrate. The substrate will contain a mixture of fecal matter, saliva, fecal fluid, and sometimes organic material as well. They let the ball dry and keep adding layers and letting it dry until it's the perfect size, around 0.8 millimeters. When this ball is the perfect size, they take it over to an established colony of fungi and they rub it on there. And then they either place it on the nest floor or they hang it from plant roots on the ceiling, and it becomes its own garden. Super interesting. Here you can see the yeast cultivars, which is the top middle here, that little green bar. That's what we just did. And now we're going to move on to the higher agriculture. This rose around 27 million years ago in a clade of 9 genera. They cultivate multinucleate or polyploid fungi, which means that they can have more than one nucleus or more than 2 sets of chromosomes. They consistently produce food bodies known as gonglidia, which we will get into, and they're obligatory. They cannot live without each other. Found across North and South America, with nests varying from 100 workers up to 10 million. The 9 genera within higher agriculture are Sericomyrmex and Trachymyrmex, which are monophyletically similar in that they sometimes are found using fresh plant material. They don't partake in leaf cutting, but they utilize fallen plant litter. Within Trachymyrmex, we had 3 different genera that were derived from it. Xerolidor explicatus, just that one species, derived in 2018. Then Paratrachymyrmex and Mycetomularius derived in 2019. Mycetoceritis, which was considered a dumpster genus of Atta, Trachymyrmex, and Scyphomyrmex from 1907 to 1950. It was a dumpster subgenus. They moved it to its own genus in 1950. Then we have Atta and Acromyrmex, which are our original leaf-cutting genera, and then Amoemyrmex, which was derived from both of those in 2020. The higher attine fungi can be divided into 2 sister clades. They both rose from lower agriculture clade 1. We have Leucocoprinus gongoliferus, that one single species, which was originally thought to be exclusive to leafcutter ants, but has since been found being cultivated by a few others. And then clade B, which contains multiple species, and usually non-leaf cutting ants cultivate this within the higher attine ants. But once in a while, leafcutters can be found cultivating clade B fungi as well. Leucocoprinus gongoliferus was thought to be exclusive to the leafcutters, but we've also seen Trachymyrmex, Paratrachymyrmex, Mycetomularius, and the lower attine species, Apterostigma megacephala. Apterostigma will consistently come up on these lists as an exception. Gonglidia. The Greek word gonglo, meaning rounded, is the inspiration for this name. It's one of my favorite words to say. It has specialized hyphal swellings that grow in bundles called staphyli. These ants suppress the sexual reproduction in the fungus. That's why they don't have these fruiting bodies that we're used to seeing. It's thought that their pruning both stimulates the formation of gonglidia and represses the sexual fruiting bodies. The quality of substrate, pH, and temperature have also been shown to affect the growth and size of the gonglidia. The gonglidia produce a lot of different nutrients like lipids, glycogen, amino acids such as arginine, and a lot of important enzymes that the ants ingest, and then they distribute onto patches of vegetation so it helps the fungus digest it. They are found mostly in higher agriculture, but some lower. Here are some exceptions in the lower Atini. Apterostigma wasmanii, Scyphomyrmex strigatus, and Mycosapirus smithii, all lower agriculture, have been found with fungi producing gonglidia. The top 4 pictures are from one of 3 of these, I think Mycosapirus. And the only difference that can be found between that and the gonglidia of Leucocoprinus gongoliferus on the bottom here is the size. So we're not sure if it is the same type of fungi or if it is its own. Some interesting facts. Only Leucocoprinus gongoliferus produces gonglidia in a culture medium. Any of these others just revert back to filamentous mycelial growth when they try to grow them in culture medium. So that kind of hints at it being a totally different type of fungi, in my opinion. Arginine. This is an essential amino acid that attine ants have completely lost the ability to synthesize. So they need their fungi to to synthesize and get this amino acid. It gets converted in the ant's body into proline, and it improves their cold tolerance by stabilizing their cell membranes. All right, so we've gotten through our higher fungal cultivars. On this chart, leaf-cutting ants is going to be under what you can see says Leucoagaricus gongoliferus. That was reclassified to Leucocoprinus in 2023. All right, leaf cutters, 18 to 19 million years ago, they rose. They're considered the dominant herbivores of the neotropics. They consume 25% of the daily plant mass. There are 52 species within 3 genera, Atta, Acromyrmex, and Amoemyrmex, and they are completely obligatory with their fungi. It says with Leucocoprinus gongoliferus, but sometimes they are found cultivating different clades of fungi. Not very often, but sometimes. They're found across North and South America with up to 10 million workers. These are going to be the biggest of all fungus-growing ant nests that you're ever going to see. These guys attain the highest level of organizational complexity found in non-human animals. The way that they work together, the way they build their houses, crazy. They have extreme polymorphism, which is when different castes of ants are different sizes. So you can see just the vast difference between their worker size. The small ants are called minims, and they're responsible for tending to the fungus gardens because they can fit within the little combs of the fungus, and they're responsible for brood care. Usually the brood is kept with the fungus, makes things a little bit easier. The medium-sized workers are called majors. The majors are responsible for all sorts of different things, from brood care. They can help with brood care sometimes. They can help in the fungus gardens. Also, they are responsible for transporting the leaves from the bottom of the tree back to the nest and then cutting them up. The giant guys here, gals, are super majors, and they have these big muscular heads. Their only job is to cut leaves. Sometimes they'll bring it back. Sometimes they'll bring it back halfway and drop it. Their main job is just cutting these leaves. Here is a picture of an Atta nest that was being excavated. They used 6 tons of cement and 8,000 liters just to fill all the chambers and tunnels. Ant nests can go as far as 20 feet deep for these leaf cutters and up to 100 feet across. It's just massive. Their nest chambers can be about the size of a human head, and they can have anywhere from hundreds to thousands of these chambers. And they're all positioned at a very specific depth to maintain specific humidity, temperature, and airflow. So this is all the relationships that we just covered. We've got the 2 different families within the order Agaricales. Got our coral agriculture. Before we move on from the different agriculture levels, I just wanted to go over the different hierarchy that you can find in the US. I comprised this list because I wanted to know where to start looking. And awesome enough, we have 6 of the 9 different higher agriculture. So we have 2 of the leaf cutters. And here's a species breakdown. So it's not so surprising that you can find a lot of these ants in the south of the US, but the northmost, Trichomyrmex septentrionalis, is also the most widespread, can be found as north as Illinois. So I'm betting that there's some in Wisconsin somewhere, and I'm going to find them. All right, so now we're going to go over the last 2 things we have to talk about, which is going to be enemies and defenses. So although people are usually the enemies of most living things, today we're going to be talking about fungal parasites. There are 5 different mycoparasites within the Hypocreaceae family that are found to kind of take over these fungus gardens. Starts with Escovopsis. Escovopsis was originally thought to be the only mycoparasite within these ant nests, but it was later discovered that it actually had three sister clades that were all mislabeled as Escovopsis. And the color of their colonies was one of the biggest differentiators. However, it was noted that they were different colors a long time ago, but the lack of morphological analysis led to an oversight in the fact that they're completely different genera. So Escovopsis is brown, Escovopsioides is white, Symphodio rosea is pink and yellow, and Luteomyces is also pink and yellow. The pink, yellow, and brown are unique to this family and aren't usually seen. Trichoderma is not related to Escovopsis, but is another mycoparasite in the Hypocreaceae family. If you cultivate mushrooms, you might know it as the green mold disease. It can be quite detrimental to the crop. They go after decaying organic material, and that is exactly what these ant nests are full of. So they can be quite detrimental. Here is a little chart showing the morphological differences between Escovopsis and its sister clades. They have different types and shapes of vesicles, phyalides, and conidia. Conidia are the spores of asexual fungi, which are essentially an exact copy of its parent. And the phyalides are flask-shaped, and they're attached to the vesicle and are responsible for helping produce these conidia. So Escovopsis has a lot more developed and elongated phyalides compared to Escovopsioides. Luteomyces produces multiple conidia within the same cell at the same time, and Symphodio rosea has a lot more branches. Escovopsis is a mycoparasite or hyphal parasite, was considered a single genus, like I said, until 2023, and now contains at least the 4 genera we went over. Most of the research done on Escovopsis until 2023 was actually Symphodio rosea. And so since they've started to look into it over the last 2, almost 3 years, it's been noted that Atta, Acromyrmex, Mycetum malarius, Paratrachomyrmex, Sericomyrmex, and Apterostigma in the lower agriculture are the only so far documented with actual Escovopsis. The rest of the clades of this mycoparasite seem to go after most of the other genera. There's so much more research to be done on that, though. Now we're going to go over their defenses against these mycoparasites. We have Actinomycetes, originally called Actinobacteria, which is a group of true bacteria that is very similar to fungi, in that they also produce a filamentous branched colony that hangs over the substrate, and they act as decomposers in the wild, degrading organic compounds. 2 different genera, Pseudonocardia and Streptomyces, have advanced into a symbiotic relationship with these ants. They are passed on both vertically and horizontally and secrete antifungal, antimicrobial compounds. It's thought, but not sure, that they absorb nutrients from the cuticles of the ants, but it hasn't been entirely proven yet. So Pseudonocardia, this is going to be the most specialized of the 2, thought to evolve with the ants over time, but is still also free living. Most of the samples taken from attine ants have been genetically identical to free-living Pseudonocardia. They can be found naturally in soil, decaying organic material, and marine environments. The only genus of attine ants that do not have this is Atta ants. So they rely heavily on their PAA secretions, which I will tell you about in just a little bit. They have antibiotic, antimicrobial compounds like dentagermicin, germicin A, B, and C, and a Pauline antifungal, nystatin. So it's really a miracle that they have developed a relationship with something that fights off fungi, but not the one that they're cultivating. The more primitive ant genera, so the older ones, They show the greatest accumulation of these bacterial cultures on the underside of their abdomen, whereas the more derived genera they've developed a special patch on their chest area that carries these cultures. The different types of casts have different amounts of the bacteria on them. The farmers, the gardeners, they are more coated, like this picture of this acromyrmex species. They're coated completely because they're working with the fungus. Whereas workers or majors or super majors have a lot smaller of an accumulation, but they still do have it. Streptomyces reacts very similar with the ants to pseudonocardia, but it is frequently adopted from the environment. It is not seen to be passed down vertically from queen to daughter. It's seen only to be taken from the environment. Also found in soils and with decaying organic material. They have the ability to produce antimicrobials that have varying levels of aggressiveness, depending on the level of infection in the nest. Some more interesting facts: ants are not born with this bacteria on their cuticle. They accumulate it over a couple of days of interacting with nestmates in the fungus gardens. The last defense mechanism is those PAA secretions. Phenylacetic acid is a chemical compound produced by the metapleural glands, which are a type of exocrine gland found only in ants. You can see in that red rectangle where it is. It's at the bottom of the thorax. It's shown that Atta and Acromyrmex have a lot larger metapleural glands, probably because they rely on these so much more than the other species. Studies show that the ants secrete onto their forelegs and then through meticulous grooming, spread it over the rest of their body. They contain acids and other antimicrobial compounds. And here is a summary of all of the fun we just went over. So we have the 5 different practices of agriculture: lower, coral, yeast, higher, and leaf. And then we have the 2 different families. Pteralaceae and Agaricaceae are the fungi that they cultivate. We went over the mycoparasites in the Hypocreaceae family: Escovopsis, Escovopsioides, Symphodio rosea, Luteomyces, and Trichoderma. And then we discussed the actinomycete defenses, Pseudonocardia and Streptomyces. And then lastly, we talked about their PAA secretions from their metapleural glands. Well, thank you. Thank you for having me on today. And here is some recommendations for some books if you're looking to read more about them. These are my favorite right now. You can find me on my social media or email me at bugswithbeth@gmail.com.
Woo!
That's what I got.
This was
Wow.
amazing. Are there any other books besides this?
Oh, there's so many books. I can email anybody. If you want to send me an email, I can send you a whole bunch of books.
Beth, I want to say I've learned about fungi cultivating ants in mycology class and entomology class, and this is more detail than and really cool like way of breaking down the information that I've ever had before. So thank you, first of all, so much.
Oh, absolutely. Thank you.
Wow. So there was one question I saw in the chat already, and if anyone wanted to either type up their question in the chat or prepare to unmute yourself and ask it after I ask this one, go ahead and do so. But this one was from Aaron, and he was asking, so like, why is it called lower agriculture and higher agriculture? Like, is it, yeah.
Yeah, so it's just lower and higher essentially because of the way that how specialized they are. So lower agriculture means that they more often are just yoinking mycelium right from the woods and growing it within their nest, but they always target the same family of fungi. So they identify it, but that usually just means that they're taking it from a free living species. They're using more ancestral substrates, so like decaying material, insect frass, that type. And then higher agriculture is usually when they're cultivating those specialized fungi that are only found in those nests. They're only passed down from queen to daughter, and they can't survive. And they usually, but not always, use fresh plant material.
Very cool. So it's like about the relationship to the fungus and how they
Yeah,
get.
whether it's more specialized or if it's more willy-nilly, as I put it.
Yeah.
Beth,
I know some human growers that differ in those two ways too.
Yes.
I have a question.
Go for it.
Beth, have you ever seen any of these ants? Sorry if I missed that if you said.
No,
Wow. Well, in La Crosse, we have a colony of Atta texana if you want to come see them.
I will be there.
In captivity, I should say.
Right.
Yes, yes, they are in our entomologist's house right now, but I believe he would be very happy to show you if we told him you're interested. I've taken care of them a couple times when he was out of town, and yeah, they're pretty voracious. Yeah, I just drop in some raspberry leaves or some orange pith and come back later in the day, and they've incorporated it. And he has little clear boxes so you can actually see them building their nest in their garden. It's really cool.
Yes, that's incredible. I would love to see that.
We should arrange a trip to the tropics too to see some of the leaf cutters because they're just like first time I saw them I just sat down on the trail and just like got eye level and recorded them with they're perfectly in a row carrying the leaves. It's so sweet and it's just like mesmerizing and we should all go hang out and look at them.
I agree. I have always wanted to make my way down there.
I agree. Let's make it a trip.
Yes. Group trip.
Perfect. I'll brainstorm.
Am I supposed to be reading the chat here?
You can. I usually try to catch them, so I can
Okay,
read
I'll
out
let you know.
a question from Levi. Have you ever gone to Illinois to find the populations of Trachymyrmex? And have you ever kept them as pets? So,
I have tried. I've gone to Illinois to look for them, but I haven't found them yet. And I have not kept them as pets, just because I've debated it, but I'm not quite where they're supposed to be. So there's been that kind of debate within myself about it, but I might maybe if I move more that way.
yeah, like so sort of the concern about accidentally having them, like
Yeah,
being the human mediated dispersal, as we say.
exactly. And they can be destructive sometimes. So no, probably not.
Yeah, don't want to be that person to your neighbors.
No. No more tree for you.
Yeah. Um, Here's a question from JMO. During the very cold times on Earth, the ice ages, it seems that ants pulled fungi from underground. Could it be inferred that ants saved fungi on Earth?
I don't think so. I think fungi has its own way to do everything. If anything, it's more hardy than ants. So I don't think so, but it's a cool thought. I think they definitely help disperse it.
Yeah, like these particular lineages, maybe, but fungi have been,
Right.
as you say,
Right.
all over the place.
Exactly.
There's almost nothing they don't eat or do.
Yep.
The ants were like, "You're mine now."
Right.
The
Like you're
fungus
right.
didn't fully have a choice in some cases.
Um, I had kind of a question about, and I know you haven't yet had success finding them, but when you go out to try to find like the tracking Myrmex, if I'm saying that right,
Mhm.
what is your method? Like, what are you?
I wish I had more of a method to say. I know that they prefer kind of more damp soils to drier areas, fungi. But other than that, I don't have any sort of specific way I go about it. I just, sometimes they make little trails and, searching for any sort of trail through the leaf litter or whatnot. But no particular efforts or strategies, I should say.
I really feel your pain as someone who's just been studying truffle fungi lately. Like,
Oh yeah, you can't even see those.
there are methods, but they're very like, well, you try this, and then you mostly just dig a bunch of empty holes.
I should get a dog.
Yeah, smell the ants. Actually, the ones in captivity we have, like, they have a very unique odor. It'd be cool to
Mhm.
be like, could you train a dog in scent work for
Yeah.
ants? Maybe. Pat in the chat here has from the Blue Ridge Mycological Society sharing his tips, I think, which is asymmetrical nest mounds and sand.
Oh, yes.
Asymmetrical nest mounds. So I guess like got kind of a
Yeah.
slope and a bump. Open areas, also here in Virginia, a more the northern population, they seem to be drawn to quarries.
Okay.
That's cool. So maybe, yeah, maybe the mycological societies from the regions where they're more common. I wonder how often
for sure.
their fungus makes the species lists. Cool. Oh, specifically abandoned quarries. We have a decent amount of them in Wisconsin, maybe to check out. Because like I was interested in saying where like they've been seen in Illinois. Was there a particular reason you thought that they might be in Wisconsin? Do you think they're like moving their range or just that they're underdocumented?
They were documented right on that line border between Wisconsin. Like it was just barely Illinois. And so it just led me to be suspicious. But, you know,
I'll have to keep my eye out because we have so many quarries in central eastern Wisconsin or closed quarries. So I'll let you guys know if I can try to look them up and see if there's anything here that would be worth exploring.
absolutely.
Well, and Pat says he's going to text us pics of the nest sites.
Oh, awesome. I can't
Yep.
wait.
Cool. He is also asking, he's saying in the fall they throw out a bunch of dead fungus and it's easier to spot. So I guess they have like a waste.
The waste pile, yes.
Yeah. And now we have a question from Max Reinhardt. And he says, not to anthropomorphize, but is there anything we can glean from these different ant practices that might inform our means of farming fungi or farming in general? Yeah, I guess like biomimicry. Can we learn from the ants?
Yeah. You know, I think that maybe we could if we kind of figured out their methods a little bit more. With the sexual like how they prevent them from having their sexual spores is still kind of a mystery. It's just lots of pruning and good pH, and I think we could with more research, but it definitely needs a little bit more just to study how they do it and why.
Yeah, like and how exactly they're utilizing some of the things you listed. I kind of like what picked my ears up was the phenylacetic acid, because like they're using that as a biocontrol, or not biocontrol. They are the bio, but they're producing as a chemical control, and it seems to be a pretty straightforward compound. Like I was kind of curious about it. Yeah. Pat says we all need to store massive amounts of bacteria under our chins.
I think so too.
We kind of do, not in a specialized organ though. JMO asks, it's a kind of elaborating on the question that was about the like surviving fungi in like cold periods or ice ages. Was like I meant pulling certain fungi underground through specific periods of ice and cold to preserve them. Like maybe those lineages perhaps were surviving underground.
Yeah, that could be.
Yeah, especially if they were adapted to warmer temperatures. It's always something to look at.
Right.
Glenn is asking, are there any associates of slime molds? So not fungi, but like we love them anyway, but slime molds with ants.
There are not, to any documented knowledge, no. Just the corals and yeasts, and then the glucoproteinaceous fungi.
Anyone want to unmute themselves and ask a question or write one?
I'll ask a question. I'm on my vibration plate, so I'm vibrating away. But Beth, other than the green mold that you talked about earlier and cordyceps, is there any other like fungi that you would consider like kind of really dangerous to ants?
Oh, the Escovopsis as well as the trichoderma. But parasite-wise, I think that there could be a whole range of just bacteria that could be transferred into their nest. That any bacteria really. They have a lot of decaying matter in there that can draw mold. So I think that they have a lot of potential parasites, but the sheer amount of good bacteria and antifungals that they carry, not only on their body, but just about their whole nest, I think is a really good preventative measure, and I think it's worked out pretty good for them. But, things can evolve, like the Escovopsis, into 3 different sister clades.
Yeah. I kind of was curious about the way that they some of not all of them, and I'm sorry I'm not remembering which one's which right now because I don't have the encyclopedic knowledge of it quite yet. But the ones that are the fungi are having their sexual cycles suppressed. I've heard of that sometimes getting brought up in other contexts as like, how mutualistic is it really? Is the benefit that the fungus gets from being cultivated good enough to be to sort of outweigh the evolutionary harm of being prevented from sexually reproducing, et cetera, et cetera? That's not really formatted like a question, but I don't know if you considered that.
I get what you're saying. Yes, I have. So one of the opposite effect is how the ants stopped being able to synthesize that arginine. I think that that is something that the fungi specifically started to do for them to make them reliant. I think that their benefit is that they just have worry-free living and reproduction, perfect temperature, perfect humidity. Doesn't matter what temperature the world gets outside. So I think that it's really just a lazy, easy way of living. And they've kind of found a way to make sure that these ants worship them essentially as well.
So domesticated, but domestic.
Exactly.
That's the view I take on lichens too, and the kind of debate over about are the algae captured and enslaved, or are they like happy in their little kind of plush prison? And I think it's too plush to be.
There's a similar debate over the bacteria, the actinomycetes bacteria, like what are they getting from it? And like I said, it's hypothesized that they might be taking nutrients from the cuticle of the ant, but it's not entirely sure. So, why are they hanging out? Because they're also just as equally free living. So I think it's just a beneficial opportunity at this point.
Yeah, like a house and
Yeah,
maybe getting dragged around into places where they have food.
exactly.
Cool. There's
And
a
then
couple.
they can feed on that organic material in the nests.
Yeah. I think that hitching a ride on various insects seems to be a really good way to get to a lot of food if you're a
Yes.
fungus or bacteria. There's a couple more questions from the chat, which Glenn asking, how much study has there been done on the antifungal chemical strategies of some of the ant genera?
The antifungal strategies. Can you elaborate a little bit more? Sorry.
Might be typing.
No, that's fine. Take his time.
Or potentially trying to unmute himself, but is not unmuted. Well, I don't know if I can prompt somebody to mute themselves. In the meantime, perhaps we could ask one of the other questions.
Yeah.
Oh, there is an elaboration I missed actually right under there. I didn't see it. Particularly antifungal strategies that might become a medical strategy for mammals. So I think correct me if I'm wrong, Glenn, but basically, have we looked into whether these antifungal chemicals would be useful for human medicine or other mammal medicine?
I want to say that they have asked this question before, but I'm not personally familiar with the research done on it. I haven't fallen down that side rabbit hole yet. But that is a really good question and definitely something I would wonder too.
Yeah, my experience reading up about other antimicrobials is we're desperate enough for more of them that if there's one people know about, somebody's trying it.
Exactly.
And it takes a long time to get those through the approval pipelines. So that's usually the hang up. probably definitely something to research more. There is a question from JMO. Is there a perpetuation of certain fungi directly linked to ants?
As in only with the ants?
Yeah, I guess certain fungi being grown or spread by them. You could elaborate if you want, JMO.
So, the Leucoprinus gongoliferus is strictly grown with the ants, if that's what you're wondering. But that is the only one with a name that is exclusive. The rest are, mystery A, B, clade C.
Cool. Do you get the sense that I'm guessing the answer is just yes, but that there's a lot more to be done with the taxonomy of some of these like fungal species that are grown with the ants?
Very, very much so, yes.
Yeah.
There was a lot of ends left open, but it's a constant, in any science, just looking for answers and different approaches to the research you're doing.
Okay, any other questions people have?
Yeah, Mariah, when's this going to be available on YouTube?
So we've downloaded it to the cloud, and I think we're just going to try to put it up right away because I know multiple people who want to see it. So, and we're trying to get better about that, like getting them up.
NAML wants to see it. I had a meeting about that last night, and they want to view it immediately for speakers in Michigan.
Wow. Okay. Well, let's get it up tonight then.
Okay. Thank you.
We do have a question. More people thanking you and saying great job, Beth. But also, have you seen much interaction with ants at the roots of plants where mycorrhizal fungi engage with them?
There hasn't been any notes or documentation that I've seen of any particular plant being required. They usually grow kind of farther away from plants, but sometimes they grow near them, but there aren't any that are specific or repetitive enough.
Yeah, that would be. I mean, like every time there's a connection with fungus and an ant or like a insect, you always like, who else is going on here?
Exactly. What's living near this?
Yeah. And multiple people are saying, thank you. What a wonderful summary of this topic. This was fascinating. Thank you so much. So that's my feeling too.
Well, good. I'm glad. Thank you.
Yeah. And please do come visit Lauren and me in La Crosse to visit the ants.
I sure will. I promise.
And I think Lauren was advertising going to the New Jersey Pine Barrens to look for the Trachymyrmex septentrionalis.
Yes, I would love to find them. I have a appointment in March with the American Natural History Museum to see their colony in New York.
Wow.
So I'm excited for that one.
And, a question from Levi. I have seen lots of root aphids that ants cultivate underground. They can sometimes look like fungi because of their fluffy bodies.
Oh,
Maybe
yes.
the question is a report from Levi.
Yes, they do. The little fuzzy aphids are my favorite.
Yes, and you're being invited to Virginia to see.
I'll go there too.
Yeah. Make a big
Here I
tour.
come, guys. Here I come.
Yeah, and no pressure if you don't want to, but we would love to see any pictures or experience or written experiences from your trips to see these fungi cultivated ants for the newsletter.
Oh, for sure. Yes, I will definitely be documenting everything.
Mm-hmm. Okay, so any last questions from people? Jeremy is about to go fill his intrabuccal pocket with cookies. I think we should all call our cheeks intrabuccal pockets.
Yes, yes. It's a good storing system.
It really is. Thank you so much, and I'll get on uploading this to YouTube right away for the people.
Awesome. Yeah, thanks
They
for having
know.
me. They gotta know.
Good night, everyone, and welcome again, Beth and Danny, to the Debbie Must board.
Good night. Bye.
Good night. Bye.